Hello, and thank you all for joining us today for this MBL International webinar hosted by Samba Scientific. My name is Jason Amsbaugh, and I'll be the moderator today. In this webinar, Dr. Yuri Polektov, an R&D scientist from MBL International, will present data on their QuickSwitch MHC tetramer platform and how it is used to quickly make unique tetramers and measure the strength of peptide bonds. Without further delay, I'll turn the time over to Dr. Yuri Polektov.
Okay. All right. I hope everyone can see my screen.
Looking good.
Okay, everyone can hear me? Okay, my name is Yuri Polektov, and I'll be talking to you about making custom MHC tetramers at the lab bench using the QuickSwitch kit. Oh, sorry. There we go. Oh, wait. Let me just... There we go. Okay. Tetramers are currently the most common method for identifying individual T cells. Unfortunately, the interaction between a T cell receptor and a peptide MHC molecule is relatively weak and only lasts for a few seconds. This interaction can be improved by coupling a number of MHC molecules together so that when one peptide MHC molecule dissociates from the T cell receptor, another one takes its place and the binding lasts a little bit longer. Using this approach, 4 MHC molecules were bound together with an avidin molecule, and the first peptide MHC-based staining of T cells was performed by the Altman lab in 1996. Since then, tetramers have become commonplace in any lab trying to identify a specific T cell population. Tetramers for both class 1 and class 2 molecules have been developed over the years. Due to their functional and structural differences, class 1 molecules generally stain better than class 2, but class 2 molecules identified some of the more widely studied CD4 T cell populations. Class 1 molecules, for the most part, remain stable for as long as they're bound to the peptide and fall apart as soon as the peptide dissociates. Class 2 molecules are usually synthesized with a leucine zipper that keep the alpha and beta chains together in a synthetic MHC molecule, and they can still remain stable even if the peptide dissociates. But due to the structural differences, the class 2 MHC molecules are a little bit more promiscuous to the binding of various peptide sequences than class 1 molecules. While the primary use of peptide MHC tetramers was and still remains the staining of T cells using flow cytometry, there are numerous other applications. Tetramers can be used to detect live T cells using confocal microscopy. And if you look at the picture on your left, this is an example of an experiment where MHC class 1 tetramers were used to visualize CD8 positive T cells. Using more complicated microscopy techniques, it's actually even possible to track cells in real time as they move around the body. And in some cases, MHC tetramers bound to beads or other scaffolds can be used to stimulate the right population of T cells. But for all those techniques to be effective, the effectiveness of each tetramer used is limited by the nature of the peptide that's loaded into the MHC molecules. Some peptides bind with high affinity to their MHC molecules and have strong T cell targets that produce great staining results, while others do not. In order to determine which peptide can cause the best T cell response, one would have to test numerous peptide sequences, sometimes covering the entire protein, and that requires a lot of different MHC tetramers. Traditional methods for making new MHC class 1 tetramers involves the refolding of synthesized MHC class 1 heavy chains with beta-2M and the peptide to be loaded into the tetramer. This traditional method can add a week or more to the production procedure in addition to the time it would take to synthesize the peptide and test the newly folded MHC molecules. Taken together, it could take up to a month between when you place your order for an MHC tetramer and when you can actually get it in the lab to start your experiments. This delay does not allow for changing your experiments based on intermediate results, and it requires the researchers to know exactly what peptides loaded in what MHC alleles are necessary for the experiment before it even begins. The addition of every new tetramer will significantly increase the cost of the experiment, and it will also leave behind a lot of unused tetramer when you're done. Another approach for making MHC tetramers involves the ability of MHC molecules to exchange the peptide that it was loaded with for another peptide. This is something that if any of you work on basic science or MHC research, you've probably done in your lab. Like that, this happens all the time. Peptides get exchanged even on the surface of cells. We have developed a system that could utilize this ability of MHC molecules to exchange peptides, allowing our customers to create as much tetramer as they need, loaded with whatever fitting peptide they desire within hours as opposed to weeks. This way, you just have to wait for your peptide to be synthesized, and once you have it, your tetramer can be ready to stain cells in less than a day. No matter how the MHC tetramer is generated, if all the proper purification procedures are followed, the final tetramer product works the same way when it comes to T-cell staining. This is an example of Jurkat Mark I cells that are stained with the Mark I PE tetramer, either in a QuickSwitch procedure or a Mark I PE tetramer that's been refolded using the traditional method. And as you can see by the MFI and just by even the size and shape of the graph, the staining is almost identical. Our system for generating MHC tetramers through peptide exchange is called QuickSwitch. Through multiple testing, we have validated that for most MHC molecules, the peptide exchange principle applies even when the MHC molecules are already bound to a streptavidin molecule or tetramerized. And this is our little cartoon of a tetramer. By loading the MHC molecules with a weak binding peptide, or what we refer to as the exiting or the QuickSwitch peptide, and adding a special peptide exchange factor, we're able to complete the exchange reaction in about 4 hours. The one major drawback of creating tetramers using the peptide exchange procedure is that if the peptide you're trying to load is not a good binder, there will still remain a lot of unexchanged exiting peptide in the tetramer. For this purpose, the QuickSwitch system comes with all the necessary components to measure how much of the target peptide is loaded on the tetramer and how much of the tetramer still has the weak binding exiting peptide still bound to it. The QuickSwitch MHC Class 1 kit includes the following components. First of all, the exiting peptide loaded MHC tetramer solution in the kit is supplied at 50 micrograms per mil. That's about 1 micromolar of MHC. And when I say 50 micrograms per mil, I mean just the concentration of the MHC, not the streptavidin or the fluorophore. And there's enough of it for about 10 exchange reactions, each resulting in 50 microliters of tetramer loaded with the peptide of your choice. So about 50 microliters of 1 micromolar MHC in solution. That's how much per experiment you will have. The FITC labeled antibody included in the kit is specific for the weak binding QuickSwitch or exiting peptide. Each kit comes with a small aliquot of a strong binding peptide that is guaranteed to exchange nearly all of the exiting peptide in an exchange reaction. And this peptide can also be used as a reference peptide for the exchange procedures, as well as a negative control for the tetramer staining of cells. Each MHC class 1 kit comes with magnetic beads that are coupled to an antibody specific for the MHC molecule of the kit. This allows the beads to bind the MHC tetramers properly with properly folded MHC molecules independent of what peptide is. Volume of PAF for about 10 exchange reactions in each kit. And please do not attempt to perform the exchange reaction without adding any PAF. Even if you're sure that your peptide is a high binder, it will not work. All the necessary volume of wash buffer that will be used for the wash of magnetic beads and the dilution of the anti-exiting peptide antibody is included in the kit. In order to make the MHC tetramer with the peptide of your choosing, you have to mix together 50 microliters of tetramer with 1 microliter of the peptide exchange factor and 1 microliter of peptide diluted to 1 millimolar in DMSO and H2O. For most MHC class 1 tetramers, you can have an effective exchange with only using 20 micromolar of competing peptide in the final concentration in this final reaction. And the final concentration of DMSO is going to be about 10% in the peptide solution. That's usually what you're going to end up with. But the exchange reaction can take place, it can handle up to 10% concentration of DMSO in the final exchange reaction without any difficulty. Let the peptide exchange take place for about 4 hours at room temperature. And at this point, the tetramer is ready for cell staining. However, if you want to know the effectiveness of the peptide exchange, then you have to proceed to the next step. So this is an example of stimulated PBMCs that are stained with HLA-A11 MHC tetramers, either folded traditionally, just off-the-shelf tetramers that we have that you can buy from our company as well, or with the QuickSwitch exchange. And as you can see, using a peptide stock of 1 or 10 millimolar for the QuickSwitch exchange reaction does not change the staining results. So first of all, the results are almost identical with the tetramers that are generated through refolding as opposed to peptide exchange. And no matter whether you use 1 millimolar or 10 millimolar in the peptide stock, it's still the same. To determine the effectiveness of the peptide exchange, incubate 5 microliters of the exchange tetramer from a total of 50 with 20 microliters of magnetic beads while shaking the plate at 550 RPM for about 45 minutes. Our kit was developed with the intention of using a 96 well V-bottom plate for high throughput screening and peptide staining procedures, but it can work with using individual tubes. So if you only have 1 or 2 samples, you can just do this in a tube. You don't have to go through a plate. Next, dilute the wash buffer provided in the kit 1 to 10 in water and use it to wash off any unbound tetramers. For this process, we recommend using, once again, the magnetic plate to bind the beads on the bottom of the plate, but it's possible to simply spin the plate at 350 Gs for 5 minutes to have the beads precipitate in the bottom of the plate. The magnetic beads are heavy enough, like, yes, they're magnetic and you can have other things that you can do with them, but just because of how heavy they are, they will precipitate in just a regular centrifugation. So you don't really need the magnetic plate if you don't want to use it. With the magnetic plate, it's going to be a little bit better and cleaner. When you can then use a strong hand motion to simply flick the liquid from the top of the beads out of the plate, leaving behind the beads alone. Just like, if you guys have stained cells, I'm sure you know exactly what I'm talking about. Just flick it into the sink and the beads will be on the bottom of the plate. They will not, I mean, if you spun it with at least 350 Gs for 2 minutes, you'll be fine. The beads will remain in the plate. Dilute the provided anti-exiting peptide antibody in the wash buffer and add 25 microliters of this antibody dilution directly to the beads at the bottom of the 96 well V-bottom plate. Shake the plate at 550 RPM for 45 minutes while covering the plate with aluminum foil to prevent the degradation of the FITC signal. Using the same procedure with the magnetic plate, use the wash buffer provided with the kit to wash off any anti-exiting peptide antibodies that did not bind to the tetramers. And once the wash is complete, fill up every washed well with about 150 microliters of wash buffer and proceed to reading the beads in the flow cytometry on the flow cytometer. This entire process is described in our kit's manual. While the tetramer molecules are too small to give a forward and side scatter in the flow cytometer, the magnetic beads are not. Whatever instrument you use, you should be able to see a nicely defined population of magnetic beads that stands out from the background in its forward and side scatter parameters. Using the flow cytometer to determine, use your flow cytometer to determine the relative FITC, or in most cases FL1 signal of this population of magnetic beads, and make sure to compensate the FITC signal with the signal of the fluorophore attached to the tetramer. In our case, it's going to be PE, APC, or brilliant violet. To properly quantify your reaction, you also need to use 5 microliters of the unexchanged tetramer as a negative control, like this. And in addition, you also need to add a sample of beads only as a positive control. And let me explain how that works. The reason for using the unexchanged tetramer is important for this procedure is that since all of the MHC molecules still contain the exiting peptide, it will have a high FITC signal, and it will refer to 0% peptide exchange. The beads alone, on the other hand, without any tetramer bound, will have no FITC signal. And just like you would see if the exchange of the existing QuickSwitch peptide was performed to completion. So this would be 100% peptide exchange. The FITC signal of the peptide loaded in the QuickSwitch tetramer will probably fall somewhere in between those two values. And under normal circumstances, you probably should exchange everything and it will look more like this and less like this. By using the relative MFI FITC staining signals of the sample and the positive and negative control, you can determine the percent of exiting peptide that is still bound to your MHC molecule following the peptide exchange. What is even more important, using that same calculation, you can determine the relative percent of exiting peptide that has been displaced by the loading peptide. And this value can be used as an indicator for the peptide binding affinity. To assist in the calculation of what percent of exiting peptide was exchanged for the peptide used with the QuickSwitch kit, we recommend that you use the QuickSwitch exchange calculator spreadsheet found on our website. You just have to add the MFI values for the FITC signal of the unexchanged tetramer and the MFI values for the capture beads only to the table for the calculations to work. If any value for your experimental exchanges is lower than the exchange value of the beads alone or higher than the exchange value of the unexchanged tetramer, the table will return false as a result, right here. The exiting or QuickSwitch peptide exchange percentage can tell us a lot about the peptide's binding or loading affinity for the MHC molecules. if we compare the percent exchange for each peptide against the theoretical binding score for the MHC molecule provided by the IDB website, we can see that the peptides that exchange nearly all of the exiting peptide have a lower theoretical IC50, so they're predicted to be better binders. As you can see, there's a relatively good pattern going on here. While the MHC class 1 molecules generally seem to have a good correlation between their percent exchange and their theoretical binding affinity, even in a small set such as this, there are a number of outliers outlined right here. Generally, while our system is just a model for MHC class 1 loading using recombinant proteins, and it is by no means a perfect simulation of the in vivo process, we like to put our trust in peptide binding affinity experiments with actual MHC molecules, either in solution or on the surface of cells, and not theoretical calculations. But we do hope that one day the theoretical prediction algorithms become much more efficient, and there will be less outliers between our results and IDB predicted values. This is a graph of the same results, except for in this case, we use the IDB theoretical binding score as opposed to the theoretical IC50 values. And this is actually the score that the IDB website recommends people to use for their prediction. This makes the correlation between the QuickSwitch peptide exchange percentage and the theoretical binding a little bit better, but as you can see, outliers still persist. It has been shown that MHC molecules bound with that bind with higher affinity peptides are more stable to temperature-induced denaturation, and using a Cipro-orange DSF dye and an RT-PCR instrument, we were able to determine the relative melting temperature of our peptide exchange MHC molecules. As you can see, there's a strong correlation between the percentage of exiting peptide exchange for the QuickSwitch loaded peptide and the melting temperature of the MHC molecule loaded with this peptide. But there's still some outliers present, which I'm not sure I can explain, but experimental results. Here's a general flow chart for the process of making QuickSwitch tetramer and testing the affinity of the loaded peptide. With experience, this whole process can be completed in under 7 hours, but we actually recommend splitting it over 2 days. Only the first 2 steps are necessary to make a functional class 1 peptide MHC tetramer for cell staining. So like I mentioned before, this incubation of once you add your peptide and the peptide exchange factor, normally it's only necessary to do this for 4 hours. However, I would recommend, just so you don't overwhelm yourself in a 1-day experiment, before you leave the lab, just set up an exchange. I mean, just add the 2 solutions and just come back. The next morning, just let the exchange go overnight. Basically, it's going to be easier on you. It's going to be better for the peptide. It's going to be better for the MHC molecules, and just easier all around. So the steps involved in determining the binding affinity of the peptide for the MHC molecule involve capturing the peptide exchange tetramer with magnetic beads, washing the magnetic beads with the bound MHC tetramer, staining the MHC tetramers with bound magnetic beads with the anti-QuickSwitch peptide antibodies. Once again, washing the magnetic beads with bound stained MHC tetramers, and finally, reading the MFI of the FITC signal of the stained MHC tetramers bound to the beads. Once you, it's a fairly simple process. There's only like 2 incubations and 2 washes. Once you've done this once or twice, you'll be an expert at this. In addition to the already available MHC class 1 QuickSwitch kits, we've also developed the MHC class 2 QuickSwitch kits. The class 2 kit, unlike the class 1 kit, comes with a biotinylated MHC molecule at 100 micrograms per mil that are not bound to the streptavidin molecule. However, each kit comes with enough volume of empty streptavidin fluorophore for 10 exchange reactions, each resulting in, just like in the class 1 kit, 50 microliters of tetramer at 50 micrograms per mil, or 1 micromolar MHC, and 1 micromolar concentration of MHC, loaded with the peptide of your choice. The streptavidin fluorophore in the kit is specifically concentrated to work with the biotinylated MHC molecules of the kit. So if you're trying to use it with a different kit, you may end up with having free MHC molecules or free streptavidin molecules floating in solution, and that would actually diminish some of your staining results. Just like with the class 1 kit, the FITC labeled antibody supplied in the kit is specific for the weak binding QuickSwitch peptide, but in this case, it does not have to be diluted. The kit comes with a small aliquot of a strong binding peptide that's guaranteed to exchange nearly all of the exiting peptide in an exchange reaction. So for the class 2 HLA-DR kits, we use the clip peptide. If any of you have worked with MHC class 2 DR1, I'm sure you recognize this sequence. In addition to being a reference peptide to determine the effectiveness of the peptide exchange, the clip peptide provided in this kit can also serve as a negative control for cell staining, which some of you will have probably done as well. Class 2 kits, unlike Class 1, use magnetic beads with immobilized streptavidin and not an antibody specific for the MHC molecules. So please do not use the beads from the Class 1 kit for the Class 2 kit reactions and vice versa. Each kit comes with the necessary volume of wash buffer that will be used to wash the magnetic beads during the staining procedures. And unlike the Class 1 kit, there is no peptide exchange factor necessary, but we strongly recommend that you add an emulsifier to the reaction to prevent MHC molecules from aggregating with each other. And finally, the Class 2 kit comes with a volume of neutralizer since the exchange reaction takes place in a slightly acidic environment, and that may affect your staining. It is recommended to neutralize your exchange reaction before tetramerizing the MHC molecules. In order to make the MHC Class 2 tetramer, with the peptide of your choosing, you have to mix together 25 microliters of MHC Class 2 monomer with 2 microliters of emulsifier and 3 microliters of peptide of your choice, diluted to 1 to 10 millimolar in HDO and DMSO. The range here is a little bit better, different because I got to say, for Class 2 molecules, they're a little bit longer and they tend to be a little bit more hydrophobic than Class 1 molecules. So we kind of leave this up to the people using the kit. You guys will probably know your peptides better than we do. And to minimize the tetramer aggregation for some of the more hydrophobic peptides, we do not recommend using more than 100 micromolar of peptide in the final reaction volume. But some tested peptides, like the clip that's provided in the kit, can be used even at a 1 millimolar level as a final reaction, in the final reaction, without any adverse effects. Unlike the Class 1 kit, you're going to have to incubate this exchange reaction for at least 16 hours at 37 degrees. And at this point, the MHC molecules have been exchanged and can be used for other applications that do not require the generation of a tetramer. Each MHC Class 2 kit molecule in our kits is biotinylated. So if you want to coat, let's say, a well in a 96 well plate with immobilized streptavidin, then you can use this monomer for that purpose. But if you want to make an MHC Class 2 tetramer, then you have to proceed by adding 10 microliters of neutralizer and 10 microliters of fluorophore-coupled streptavidin to the 30 microliters or so of exchanged MHC Class 2 monomer solution. And after the addition of those two components, the MHC Class 2 tetramer is ready for staining cells. The fluorescently labeled streptavidin solution included in the kit is specific for the lot of the monomer that comes with the kit. So please do not try to use the fluorescently labeled streptavidin solution from one kit for an MHC Class 2 monomer from a different kit. While you will most likely get some MHC tetramers, your tetramer solution could contain a lot of free MHC Class 2 monomers and streptavidin molecules, which will diminish your staining. This is an example of stimulated PBMCs stained with HLA-DR MHC tetramers that had their peptides exchanged using the QuickSwitch procedure. As you can see, using a peptide concentration of 1,000, 100, or 10 micromolar for the QuickSwitch exchange reaction does not change the staining result when high affinity peptides are used for loading the MHC molecule. So the peptides that seem to stain equally well when they're used at 10 micromolar, even 1 millimolar, and the peptides that don't. Just like with the Class 1 kit, it's possible to quantify how much of the exiting peptide has been exchanged following the exchange reaction with the Class 2 kit. Unlike the Class 1 kit, the percent of exchanged exiting peptide will be determined on the monomer and not the tetramer. Incubate 1 microliter of the exchanged MHC Class 2 monomer with 20 microliters of magnetic beads while shaking the plate at 550 RPM for 45 minutes. The washing and antibody staining procedures for the Class 2 kit are nearly identical to those of Class 1, and you just have to use different kit components. By looking at the FITC signal of the unexchanged MHC Class 2 monomer, you can identify what percentage of MHC molecules still contain the exiting peptide after the exchange QuickSwitch procedure. The same QuickSwitch peptide exchange calculator spreadsheet that's used for the Class 1 kit can be used for the Class 2 kit to determine the percentage of exiting peptide exchanged for the loading peptide. So, this process is almost exactly the same as the Class 1 kit. It's just that you have to use different beads and a different dilution of antibody. And that's about it. With the class 2 kit, class 2 molecules, it's harder to make any statements on the correlation between the theoretical and the practical binding affinity, since we generally do not have a lot of data points. But even so, there already seem to be a lot more outliers. This is probably due to the fact that the prediction algorithms still have not accumulated enough class 2 data to become more accurate. And we would like to use this illustration to caution our listeners about relying too much on the computational prediction algorithms of when trying to find high affinity MHC class 2 peptides. Here's a general flow chart for the process of making a QuickSwitch MHC class 2 tetramer and testing the affinity loaded peptide. The only real difference between the class 2 and the class 1 and class 2 kits is that the class 2 kit requires an additional step to make the final tetramer product. So in addition to the exchange, that's going to take place at 37 degrees, a little bit longer than the class 1. You also have to, once the exchange finishes, add the neutralizer and tetramerize your exchanged MHC molecules before it's ready to stain. And the steps involved in determining the binding affinity of a peptide for the MHC molecules are just like with the class 1 kit. First you capture the peptide with the exchanged MHC molecules on the magnetic beads. Then you wash the magnetic beads with the bound MHC molecules, stain the washed MHC molecules with bound with anti-QuickSwitch peptide antibodies, wash the magnetic beads bound with stained MHC molecules, and finally read the MFI of the FITC signal of the stained MHC molecules bound to the beads to tell you the percentage of MHC molecules that still have the QuickSwitch peptide in them. As a summary of the differences in class 1 and class 2 kits, I want to note the following. With the class 1 kit, cells can be stained immediately after the peptide exchange procedure. And for the class 2 kit, the tetramers still have to be made from the peptide exchange MHC molecules before staining cells. The class 1 kit requires a peptide exchange factor that is crucial for the exchange reaction. And for the class 2 kit, no peptide exchange factor is necessary. But adding the emulsifier included in the kit is highly recommended. The class 1 kit can complete the exchange reaction in 4 hours, although once again, I recommend you do it overnight if you can, while the class 2 kit requires at least 16 hours incubation at 37 degrees. The class 1 kit uses anti-MHC antibody beads for the capture assay, and the class 2 kit uses streptavidin beads. And unless you order a special monomer kit for the class 1 MHCs, you will have to use the MHC molecules in the form of a tetramer. With the class 2 kit, once the MHC molecules are loaded with the right peptide, they can be immobilized to any surface or scaffold that has streptavidin. Both kits produce a final concentration of MHC molecules in your peptide MHC tetramer of about 50 micrograms per mil, or about 1 micromolar, slightly higher than 1 micromolar. Both kits come in PE, APC, and brilliant violet fluorophore types. And some of the kits sold have different incubation times and temperature designed for a specific MHC allele. So please be careful and check the instructions before performing the peptide exchange reaction. Other labs have successfully used our QuickSwitch kits to stain T cells in numerous applications. In all of the above examples, the QuickSwitch platform was used as a means to generate tetramers for cell staining, just as the kit was primarily intended for. But there are other uses that the QuickSwitch kit could be used for. To this end, here's a list of QuickSwitch tetramers and MHC molecules that are currently available on our website, but we're constantly working to add new MHC types to our existing product line. So this is only the QuickSwitch. So if you are interested in the QuickSwitch, if you are interested in just getting tetramers, we have our list is a little bit bigger. We have more alleles available for that. And with that, I think I'm gonna take questions if anyone has any.
Terrific. Thank you, Yuri, for that presentation. The first question is, can I try more than 10 peptides?
Actually, yeah, I'm just gonna. So basically, the kit is designed for 10 peptides. If you want to make 50 micro, so if you are okay with having less than 50 microliters of reaction, then you can squeeze in more than 10 peptides. It's just that we cannot be sure how well your cells will be stained with less than 50 microliters of reaction. So this is going to be up to you. The kit is designed for 10 peptides, but you can probably, if you're okay with having lower volume, you can probably do that. Bead particle size, it's about 5 microns. Sorry, no, sorry, 5 nanometers. 5 nanometers.
Okay. Here's
Uh,
another question,
I'll
Yuri.
type this.
How So one question for you.
Okay.
What if the allele that you're interested in isn't offered for QuickSwitch?
You'll have to wait. I'm sorry. It's not a simple process, and not necessarily like the discovery itself. It's just the testing to make sure that it works in multiple conditions. We're working on it. I know that some people are asking for other alleles, and it's not, it's just like it's a difficult process. Uh,
Okay.
so how does the peptide exchange factor cause exchange? That's yeah, that Okay, I can't answer that, but can the peptide exchange factor be used to make an empty tetramer? Mm, honestly, I don't think so. I don't, you won't, if you incubate the MHC molecule in solution long enough, it will dissociate, but the peptide exchange factor will probably not help you with that. That's kind of a separate process.
Okay. The next question is, can we use 2 to 3 peptides in one vial and try to check the competitive MHC peptide binding?
Um, that is a very good question. I'm not going to type this, but so when you use more than one peptide, the 2 or 3 peptides that you're using in a vial, they're going to compete with themselves. And I am not sure which one of those peptides will win. Like, yes, chances are you're going to have some of this peptide, some of that peptide, but there's also a very good chance that only one of the 2 or 3 peptides will win out in that competition assay. And you may end up with just, when you think you're going to be loading 2 or 3 peptides, you're only going to load one. So I cannot speak for all peptides and all reactions, but I just want you to be careful of that possibility.
Fair enough. All right.
So,
The next question is, what's the durability of exchange class 1 tetramers? Can I freeze them to increase durability?
you should keep them at 4 degrees. Do not freeze tetramers. Yes, some peptide. So normally we with experiments we've conducted, the kit should be stored at 4 degrees. The tetramer. You shouldn't freeze them. Once you freeze them, it usually does worse. But that's not true for every single peptide. If your peptide is a good binder, maybe you can freeze it at -20 and it will still be okay, but we do not recommend it. I think the expiration dates are, depending on the MHC molecule, 1 to 2 years. And that's the expiration date doesn't change once you switch out a peptide for the QuickSwitch peptide for another peptide. If anything, I think the other peptide will probably be more stable than the QuickSwitch peptide, so it will increase it as opposed to decreasing it.
Okay. So there was a related question, which was simply how long is that stable for after you make your tetramer using QuickSwitch?
I think the same thing. Just as long as the expiration date isn't it's not expired, your QuickSwitch, your exchange tetramer will still be fine.
Okay, terrific. The next question is, does peptide concentration affect MHC peptide binding affinity? And is it a good idea to use higher concentration for lower binding peptides?
Yes, it does. For like I showed you in most cases, it doesn't matter. You can have a whole range, but yes, there are some difficult peptides that don't like to bind, and for those we would recommend using a higher concentration. Like sometimes you want to push it a little bit. Like even with class 1, if let's say you follow the kit's procedure and you use 20 micromolar in the exchange reaction and it doesn't work, use 100. Maybe that will push it to success. Maybe only 50% exchange, and then you use 100, and then 75% exchange, and that will be better. Yes.
Excellent.
Sorry.
Next question is, can we use tetramer staining for multiplex immunohistochemistry as well? So say they have a tetramer only for CD8 specific T cells. Could they use that tetramer for CD8 in combination with CD3, granzyme B, interferon gamma, and TNF alpha regular staining?
I think, it's just a tetramer like any others. So I think if you just, if you have antibodies against CD3 and granzyme B and all that other stuff, absolutely. It's just like your regular staining panel with the regular colors. I don't think there's going to be any difference between QuickSwitch and, I showed you the results. QuickSwitch regularly folded tetramer works the same way, for at least for all our, in all experiments that we've conducted.
Okay, very good. Thanks. Next question, if why does MHC2 need the leucine zipper linker, but MHC1 does not?
Well, MHC class 2 has an alpha and a beta chain, and they're both the same. It's just they're both the same size. MHC class 1 has a very, very heavy alpha chain, and the beta chain is micro, hence it's beta-2 microglobulin. It's just the way that it's structured. Like they don't need to. It's folded better. I think honestly, like that's how originally when MHC was developed, they were developed with a leucine zipper, and people kind of got stuck with it. From my knowledge, like that, I think if you don't have a leucine zipper, some MHC class 2 molecules will stay together. They won't fall apart right away, but it's just traditional. People add the leucine zipper in there, and so do we.
Perfect. I've got one more question in the queue. So
Okay.
audience members, if you guys have other questions, please ask them now. The question I have is, could this method be used to validate neoantigen in silico prediction tools?
Absolutely. Absolutely. And for that, I would actually, I mean, if all you care about is validating whether or not a peptide binds to a particular MHC, maybe don't get the tetramer. You know, we have a monomer kit. We have a kit that's basically just the MHC molecule, and you can do that. You don't need to stain cells with it. I mean, it's going to be more cost effective to do it that way. Let's put it this way.
All right, very
Is
good.
there an A3 mutation in class 1 QuickSwitch tetramer?
Yeah.
I believe so. We'll have to get back. I don't want to, but I believe so, but I'm gonna have to get back to you on that one. I'm sorry.
All right. Sounds good. All right. Nothing else coming through. Well, with that, thank you all for attending today. If you do have any questions, you can certainly reach out either via the website at mblinternational.com or sales@mblintl.com as well. Thank you, Yuri, for your presentation today. Thank you, audience, for your time. We appreciate it. All right. Well, thanks again, and have a great day, everyone.
All right. Thank you, guys.